we are doing Science 2 subject, and we'll continue with our chapter. This chapter number 16, Heredity and Variation. So I hope you remember in the previous lecture we have studied about the structure of DNA, the Watson and Crick's double helical structure of DNA. We have studied in details, right? And so the DNA is basically deoxyribonucleic acid, isn't it? It is the most important nucleic acid or the macromolecule and the master molecule of the cell. It is made up of nucleotides, right? And the nucleotides are made up of sugar, phosphoric acid, and nitrogenous bases. I hope you remember the names of the nitrogenous bases. These were adenine, guanine, cytosine, and thymine. So we have studied the molecular structure of DNA, and we have also studied what is gene, right? So now today's lecture we are going to study about RNA, right? As we have DNA, that there is RNA also. RNA, the full form of RNA is ribose nucleic acid. It is the second important nucleic acid of the cell, right? It is also made up of the same sugar phosphate backbone, ribose sugar phosphate molecule, and the 4 types of nitrogenous bases. You can see this is RNA, which is single stranded, right? RNA is a single stranded molecule, and the backbone, the sugar phosphate backbone, okay, I'll show you with the help of a pointer. See, this is the RNA molecule which is single stranded, and this is DNA. So DNA and RNA almost looks the same, but this is single stranded, and it is so the compounds are all the same. Ribose sugar, phosphoric acid, and the nitrogenous bases. But here you can see the sugar phosphate backbone is there. It's made up of sugar and phosphate, while the nitrogenous bases, the only difference are the nitrogenous bases. So here in RNA, there are 4 different types of nitrogenous bases. That is cytosine, guanine, adenine, and uracil. So we were having in DNA, we were having thymine, but here we are having uracil, right? So the 4 nitrogenous bases of RNAs are cytosine, guanine, adenine, and uracil. So here are the nitrogenous bases, the molecular structure of the nitrogenous bases. Here, what you can see. Okay, so on your screen you can see the details of the RNA. RNA is the second important nucleic acid of the cell. This nucleic acid is made up of ribose sugar, phosphate molecules, and 4 types of nitrogenous bases, that is adenine, guanine, cytosine, and uracil. The nucleotide, that is the smallest unit of the chain of the RNA molecule, is formed by the combination of a ribose sugar, phosphate molecule, and one of the nitrogenous bases. Large number of nucleotides are bonded together to form the macromolecule of RNA, right? So this is also called as the nucleic acid, ribose nucleic acid, RNA. Now, depending upon the functions of the RNA, there are 3 types of RNAs. Those are ribosomal RNA, that is rRNA, then the messenger RNA, mRNA, and transfer RNA, tRNA. So one by one, we'll study the different types of RNAs. So the first RNA is ribosomal RNA. In short, it is also called as rRNA. You must be knowing that in the cell, there is a new cell organelle that is called as ribosomes, right? So this ribosomal RNA are present in the ribosomes, and the function of the ribosomal RNA is the protein synthesis. Here you can see the structures of the different types of RNAs, the mRNA, tRNA, and rRNA. So all three of them are having different structures according to their function. Okay, so first let us see about the messenger RNA. So from the name itself, you can find out messenger RNA, that is, it carries the message from the DNA. So it copies whatever information is there on the DNA for protein synthesis. So this messenger RNA, it goes and uncoils the DNA, and it copies the information whatever is there on the DNA, and it brings back, right? And with the help of this messenger RNA, then the protein is synthesized. So messenger RNA carries copies of instructions for assembling amino acids into proteins. The RNA molecule that carries the information of protein synthesis from genes, that is DNA chain, in the cell nucleus to ribosomes in the cytoplasm, which produces the proteins, is called the messenger RNA. Right? Are you following? So, what is the function of this messenger RNA? This is also a nucleic acid, and it is called mRNA. It copies, it carries the information, it goes into the nucleus, and it copies the information, whatever is there on the DNA, and it comes back into the cytoplasm, and it gives that information to the ribosome, where the protein is going to be synthesized. Then the next is ribosomal RNA, which I was telling you. In short, is called as RNA. So this ribosomal RNA is present in ribosomes of the cell, right? So here the function is proteins are assembled on ribosomes made up of ribosomal RNA. Ribosomal RNA helps build the actual protein. rRNA puts together the amino acid messages carried by mRNA and decoded by tRNA. Right, the molecule of RNA, the molecule of RNA, which is a component of the ribosome organelle, is called ribosomal RNA. Ribosomes perform the function of protein synthesis. Right, so whatever information is brought by the messenger RNA, it is decoded by the transfer RNA, that is called as tRNA, and then those amino acids are brought to the ribosomal RNA, and the actual synthesis of proteins takes place in the ribosomes with the help of ribosomal RNA. So this is what is the transfer RNA, tRNA. So you can see the structure of the transfer RNA. So here on the down end, there are anticodons. That means it decodes the information from the messenger RNA. And then on the other end, whatever, according to the information, whatever proteins, whatever amino acids are required, it carries those amino acids to the ribosomes. So that is why it is called transfer, because the function of transfer of the RNA is to carry the amino acids, to transfer the amino acids to the ribosomes. The RNA molecule, which according to the message of the mRNA carries the amino acid up to the ribosomes, is called as transfer RNA. Okay, so these were the three types of RNA. So I'll repeat so that you remember. So what is RNA? RNA is the ribose nucleic acid. Depending on the function, there are three types of RNAs. That is messenger RNA, then transfer RNA, and ribosomal RNA. So according to the function, messenger RNA, it carries the information, it copies the information from the DNA, it gives it to the tRNA. Then tRNA, it decodes the information, and then it takes the amino acid with it, and it goes to the ribosome. And in ribosome, the ribosomal RNA helps in the protein synthesis. It joins the different amino acids, whatever is required, and the protein is synthesized. So in this way, these RNA molecules work. I hope you have followed. Now the next topic is Mendel's principles of heredity. Now, Mendel was a scientist, or he is also called as the father of genetics. So, he was the first person to tell you about heredity and inheritance. So, here you can see Gregor Johann Mendel. He was an Austrian monk. He studied inheritance of the triads in the pea plant. Pea plant is the, what do you say in Marathi or Hindi, that is called as vatana, right? Green peas which are available in the market. So he has done his studies on the pea plant. So he developed the different laws of inheritance, and Mendel's work was not recognized until the turn of 20th century. So he actually worked in 18th century, but his work was recognized in the 20th century. So he was born on 20th July 1822 and he died on 6th January 1884. Gregor Johann Mendel was an Austrian scientist. He studied the inheritance of characteristics of the pea plant. He showed that the inheritance of these characteristics follow certain principles. Later, these principles became popular by his name. Mendel's work was recognized in 20th century. After reconfirmation of these principles, the same principles now form the basis of modern genetics. Right now, his principles, the what he has suggested in 18th century, are the basis of the today's modern genetics. So he has done a tremendous work in the field of heredity. So Gregor Mendel, he carried in studies on heredity. Characteristics that are inherited are called triads. So most of the time you must be hearing the word triads. So what is triad? The characteristics that are inherited. Those characters are called as triads. Mendel was the first person to succeed in preceding how the triads are transferred from one generation to the next. Right? A complete explanation requires a careful study of that genetics. So, genetic material is transferred in equal quantity, we know, from parents to progeny. Principles of heredity are based upon this fact. If both the parents make equal contribution to the inheritance of characteristic, then which characteristic will appear in the progeny? So this was the question mark, that now if the characters are taken from both male and the female parent, then which character is going to appear in the next generation? So he has done the experiments and all that, and he has given some laws which are very true. So all the Mendel's work were based on the visible characteristics of the pea plant. It is also called as Pisum sativum. These characters are as follows. Okay, so before we proceed, first you should know these terms. Mendel, while I'll explain you the Mendel's monohybrid cross, there are certain terms which I'll be using. So you should know these terms. The first is the dominant character and the recessive character. Right, so Mendel has said that he has named a dominant character the characters which are expressed in the next generation, right? Those he called it as the dominant character, while the characters which were not appear in the next generation at all, this was called as the recessive character. Then genotype and phenotype. So phenotype, when I'll say phenotype, phenotype is the external or the visible characteristic of the organism. For example, height of the plant, color of the seed, shape of the seed, color of the flower. So these are the characteristics which are visible to us. So those are called as the phenotype. While genotype is the characters or the genes which are not visible to us, so which are present inside the cell in the DNA, but we don't know them. So that is called as the genotype, right? And then homozygous and heterozygous, these words you must have heard. The individuals with the same kind of genes are called as the homozygous, while the heterozygous organism with two different types of genes or the factors. So here I'll show you the 7 mutually contrasting visible characteristics of the pea plant which Mendel has studied. Here you can see dominant and recessive characters when you take the color of the flower. So purple color is a dominant character while white color is a recessive character. Right? Yellow seed is a dominant character, green seeds are recessive characters. Round seed and wrinkled seed. Round is dominant character, wrinkled is recessive character. Green pods of the pea plant are a dominant character while yellow pods are the recessive character. In the same way, inflated pods, constricted pods, axial flowers, terminal flowers, tall plants, dwarf plants. So tall is a dominant character, dwarf is a recessive character. So these are the characteristics which he has studied. So not only in plants, in humans and animals also there are some dominant and recessive characters. So in human beings, so brown and black color of the eye are the dominant characters, while blue color of the eye is a recessive character. Then large eyes are dominant characters, while small eye is a recessive character. Having curly hair is a dominant character, and having straight hair is a recessive character. Right? That means dominant character means which is easily transferred from one generation to another, which you can see in the next generation. Then dimple cheeks is a dominant character, and having normal cheeks is a recessive character. In the same way, being right-handed is a dominant character, left-handed is a recessive character. Then rolling of the tongue. You must have seen some of the people can roll their tongues. So that is a dominant character, while the one who cannot roll the tongues are a recessive character. Being Rh positive, Rh factor. We know that we are having our blood groups A positive, B positive, right? AB positive, O positive, that way. So being positive is a dominant character, while Rh factor negative is a recessive character. Then taster, if you are having taste on your tongues, so that is a dominant character, and non-tasters are recessive character. And having normal skin pigmentation is a dominant character, while being albino, you must have seen the albinism, the people suffering from albino, that is a recessive character. Okay, now, so this is important. Mendel's monohybrid cross. So when you'll study this cross, then we will understand how what was the work of the Mendel and what he has done in this field. And this is also important with respect to your exams. In exam also the question is asked, explain Mendel's monohybrid cross depending on the given characteristic, right? Maybe tallness, darkness, or round and wrinkled seed, whatever character is given, you have to solve the cross and you have to explain. It may come for 5 marks as well, right? Or even short answers can be asked on this topic. And the important thing is, if you try to mug up this topic, if you try to learn this topic without understanding, then it's going to be very difficult. So it will be the shortcut method is to try to understand. If you'll understand it properly, you will be able to write this answer. So, monohybrid cross. The cross with only one pair of contrasting characters is called a monohybrid cross. That means he has studied the fertilization depending on only one pair of contrasting characters. In this experiment, let us consider the characteristic plant height, right, with a pair of contrasting character, that is tall plant and dwarf plant. Okay, so you have to write in exam, you have to explain the Mendel's monohybrid cross in the tabular form, as well as you have to write the explanation in brief also, right? Whatever you're writing in tabular form, you need to explain it and write the results of the Mendel's experiments. So here in the Mendel's monohybrid cross, first he has taken a parental generation. That means he has taken two plants of the phenotype, that means the external appearance, tall and dwarf. So he has done a cross, or he has done the fertilization between a tall plant and a dwarf plant, right? And the genotype, or the factors which were responsible for their tallness and dwarfness, he called those genotypes as dominant, are always denoted by capitals. So capital T and small t. So the first generation was, yes, what he has taken was homozygous or pure dominant and pure recessive characters, right? So he has done the cross between tall and short plant. So phenotype will be tall and dwarf, and genotype will be capital T, and small t, small t. So the gametes are capital T and small t. Right? You know that during when you have studied cell division, right? So it is mitosis and meiosis. In germ cells, meiosis takes place, and in meiosis cell division, you know that the number of chromosomes become haploid, isn't it? They become half. So here the genotypes, now the T and small t will become the gametes you will get is only one T and capital T and small t from dwarf plant. So first filial generation phenotype. So when he has done the cross, so out of this gametes, so the gametes were capital T, small t, so those combined, and he has found that in the next generation, all the plants were tall, right? But the difference was, first it was the genotype was pure dominant, that is capital T, capital T. But in the first filial generation, all the plants were tall, but here the genotype is different. You can see it is capital T and small t. Right? So then what he has done is, so now he has taken the next parental generation. So these tall plants, so again he has taken the tall plants of the first filial generation with the gametes capital T, small t, capital T and small t. So both the tall plants were there with the genotypes heterozygous. Then he has a cross. Now you can see this table which has appeared. This is called as the Punnett square. And in this Punnett square, you need to write the male gametes and female gametes in this manner. In the male gametes, or one gamete will be in the horizontal manner and the other will be in the vertical manner. So capital T, small t, capital T, small t. So here you can see the combinations. This capital T and this capital T will come here. Then this capital T and small t will combine to form capital T, small t. Now here this T, small t, and this is the capital T. Here in this box, here it will combine. So capital T, small t. Then in the next box, it will be small t, small t. Can you see this t and the this small t? So in this way, you need to write the Punnett square. So we are getting four types of possible combinations. So wherever is even if there is one capital T, so the phenotype of that plant will be tall, and uh, if there are two small t's, then it will be recessive. So here you can see the combinations. So three, if out of four plants, three will be tall in the next generation also, and only one plant will be dwarf. Right? Can you see? So these were the results of the second filial generation. According to the data collected by Mendel, out of 929 pea plants, 705 were tall and 224 were short plants. Right? So again, you can see that all plants are more, but here whatever tall plants are there, those plants are, some of the plants are homozygous, some are heterozygous, and some are short. Right? So thus the phenotypic ratio of these plants are 3:1, that is 3 tall and 1 dwarf, while the genotypic ratio here is 1:2:1. That is 1 purely homozygous, that is pure tall plant, 2 heterozygous tall plants. These are tall plants, but these are heterozygous. They have the characters in them for recessive also. And one is purely dwarf plant. Right? So thus, it can be inferred that in the F2 generation, phenotypically there are two types of plants, whereas genotypically there are three types. Okay, so this is what is important here. Okay, so in the Mendel's, what is monohybrid cross? The cross between only one types of characteristic is considered, and then a cross. So tall pea plant and dwarf pea plants were used in this cross. Hence, this is called this is monohybrid. Now, hence this is parent generation P1. Mendel referred to the tall and dwarf plants as dominant and recessive respectively. The tall plants were referred as dominant because all the plants in the next generation were tall. Have you seen the result of the first generation, first filial generation? All the plants were tall. So that is why the tall characteristic is called as dominant. The dwarf plant was referred to as recessive because this characteristic did not appear in the next generation at all. So, have you seen in the next filial generation? Here, no dwarf plant at all. So, that is why it is called as the recessive character. So depending upon these observations, Mendel proposed that the factors responsible for inheritance of the characteristics are present in pairs. Right? So in 18th century only he has said that whatever the factors are responsible for the phenotype, external appearance, those are inside genotypically, they are present in pairs. So today we refer to these factors as genes, and now we are calling these factors as genes. Dominant genes are denoted always by capital letters, so you remember while writing in the exam you need to be careful, whereas the recessive characters are denoted by small letters. So in this manner only you need to explain. So phenotype, tall dwarf, genotype, capital T, small t, small t. Then after gametogenesis, the gametes will be capital T and small t. Then in the first filial generation, you can see it has become capital T and small t. That means all the plants which he has received were tall with the genotype capital T, small t. That is a mixed kind of genotype was there here. And also we have seen that in the second filial generation, when the crossing over has taken place in the selfing of F1, so both the tall plants with the heterozygous characters, that is capital T, small t. So the gametes available were capital T and small t. And we have done the cross. So with the help of Punnett square, when we have drawn, so we have seen that the characters were in the ratio 3:1, right? So here in this table, you can try to understand capital T are pure dominant characters, which mean tall plants, which are pure. While, and that is also called as homozygous. Okay, so homozygous, pure dominant, these are the same characters. While capital T, small t, these are also tall plants. These are also tall plants, but these are heterozygous, right? These are hybrid. It is also called as hybrid. So these are also tall, but they are hybrid with a mixed type of genotype. Two types of genes are present in this. Though they are tall, but they also have the character for recessiveness. And the last one, which you can see in the fourth uh, column, there is small t, that is dwarf. So these are again pure recessive characteristic, and they are also homozygous. So again, though we can see the only two types of phenotypes, that is tall and short, but you can see the genotype, that is genes. There are three types of genes, that is capital T, pure homozygous, then capital T, small t, that is heterozygous, and small t, that is dwarf, right? That is again homozygous. So the phenotype, though the phenotypes are two, and the ratio is 3:1, the genotype is 1:2:1. One pure homozygous, two heterozygous, and one pure recessive. So while you are practicing this at home, so when you will draw this table and you will read the information again one more time, you will be clear with this topic. You will understand how it is, right? Okay, so I hope all of you have followed the Mendel's monohybrid cross, right? So before we move on to the assignment work, so let us revise. So today we have started with RNA, right? We have seen RNA is a ribonucleic acid, and it is the second important nucleic acid of the cell. It is made up of ribose sugar, phosphoric acid, and the nitrogenous bases. And the only difference between DNA and RNA is the nitrogenous base that is different, that is uracil, and in DNA it was thymine. So depending on the functions of RNA, so we have seen there are three types of RNA. Which are those? mRNA, tRNA, and rRNA. That is ribosomal RNA, messenger RNA, and transfer RNA. So if you go according to the sequence, and these are the structures of the RNA. So in exam, the question is possible that write short note on the RNA and the different types of RNAs. And these are the structures of different types of RNA. So you need to draw the structure and you need to explain each and every type of RNA. So we have seen messenger RNA. What is the function? mRNA copies the information from the DNA and it brings to the ribosomes. First, it gives messenger RNA gives this information to tRNA. So this tRNA, it takes the information, it encodes the information, and then according to encoding, whatever amino acids are required, it takes the amino acid to the ribosomes, and the ribosomal RNA does the protein synthesis. Then also we have studied about Mendel. The full name is Gregor Johann Mendel, and he has done his work on the inheritance of the triads, the principles depending on the inheritance of triads, how the characters are expressed from one generation to another. And he is also called as the father of genetics. So he has done most of his studies we have seen on a plant, that is pea plant. The scientific name is Phytotrichum. So he has done many different crosses on these mutually contrasting characteristics. So we have seen all the different characteristics. Also, we have seen the dominant and recessive characters in human beings. Then monohybrid cross, this is important. Even if you go in 11th and 12th standard, you need to know this. This is the basic. So directly considering that you know monohybrid cross, the professors in the colleges, they start explaining you in details, right? So you need to know this. So your work is you at home, whatever I have taught you today, read that much chapter carefully and try to solve this cross, monohybrid cross, whichever that is given in the textbook of tall and dwarf plant. So the cross with only one pair of contrasting characters is called monohybrid cross. We have seen the cross between tall plants and small plants. So in this manner, you need to write tall and dwarf. Then the first generation, they were all tall. Then again selfing, here's the selfing, that is crossing over or fertilization between the first filial generation, heterozygous, and then he found that in the next generation, he has also found the dwarf plants. Right? So sometimes we see in our life also, sometimes we see that even if the parents, both mother and father of the child, are black in color, still sometimes the child, one of the child, or sometimes it is the possibility the child becomes fair, right? So that means the phenotypically, though the parents, father and mother, are looking black, but genotypically they must be having the characteristic for fairness, right? And that which has expressed in the next generation. So that is also the possibility. So same can be the same possibilities are also there with the other characters. Maybe the height of the individual, tall and short, right? Even if both the parents are short, it is possible that the next generation, one of the individual, can be tall in height, even if the parents are short, right? Because though they are short in height, but they must be having the characteristic, or they must be having the genes inside them for tallness, right, which is expressed in the next generation. So that way the possibilities are there. So here you can see, as in out of 929 plants, 705 were tall and 224 were short. So in the same way, the possibility is there that the ratio is 1:2:1. So though three individual will be black, but there is possibility of one becoming pair, right? If the genotype is heterozygous in the parents' generation. So the possibilities are there. So this he has studied, Mendel has studied in the first in 18th century itself, and this was not recognized until its death. But in 20th century, his work was found, and then the scientists have studied on this, and everything whatever he has concluded were all right, and that is why the Mendel is now called as the father of genetics. Okay, so I'll give you one homework. You can take a screenshot of this. What you have to do is explain Mendel's monohybrid progeny with the help of the contrasting characters round, that is capital R, and wrinkled, that is small r, seed by Punnett square method. Right? In the same way as I've taught you right now, in the same way you have to write the Mendel's monohybrid cross using the dominant character round seed and the recessive character wrinkle seed, right? So I'll tell you in short what you'll get when you will do the cross in the first filial generation. All the seeds will be round because R is the round is the dominant character, and then in the second filial generation, when you'll do in the selfing, then you will find that it will be 3:1 ratio, that is 3 round and 1 wrinkle. So the same way, only the characters will be different, but all the principle will be same. And also you can practice the diagram of the structure of DNA. This is an important diagram in this, in our 9th standard also, for the exam, as well as for your future studies also, right? So you should know the structure of DNA, the normal double helical structure as well as the molecular structure. So this is your homework. Okay, then here we'll stop. So if you're having any doubt, you can ask me. Hello. Yes, Jainab. Hello, Jainab Sayed. You have raised your hand. You can ask me your question. Hello. Hello.
Yes, teacher. Hello.
Hi, sir. I finished with the lecture. If anyone wants to ask something, if they are having doubt, they can ask.
ओके स्टूडेंट्स, अगर किसी को कोई डाउट है सो यू कैन आस्क. अपना जो हैंड रेज करके आप टीचर से सवाल कर सकते हो. यस, सानू मिश्रा फ्रॉम पहाड़ी स्कूल. बेटा अनम्यूट योरसेल्फ.
Ma'am, what is the perfect definition of the ribosomal RNA? Hello?
Sorry, I think you have asked the question. What is the definition
morning,
of
ma'am.
ribosomal RNA?
Good morning, ma'am.
Yes, Sanu.
Haan, ma'am.
Haan.
Ma'am, what is the perfect definition of the ribosomal RNA?
Okay. I cannot hear you properly, but still, I think you're asking what is the definition of ribosomal RNA.
Yes, ma'am. Haan.
So the RNA which is present in the ribosome and which helps in the function of protein synthesis, that is called as ribosomal RNA.
Ma'am, this is the short definition of the ribosomal RNA.
Yes, I've told you. The
means
RNA which is present in the ribosome,
हाँ
and that's the function of protein synthesis is called ribosomal RNA.
हाँ yes ma'am.
Yeah.
Okay ma'am.
Okay.
Thank you.
Yes. Anybody else? Yes, Israel, you can ask. You can unmute yourself and ask the question. Hello.
यस टीचर आई थिंक कोई डाउट्स नहीं है, उसने अपना हाथ ये किया. हाँ, आफरीन शेख, बेटा unmute yourself and ask your question.
What is
Yes, Afrin.
what is phenotype and genotype?
Yes, Afreen. Mute yourself, I'll tell you. You've asked what is phenotype and genotype, right? So phenotype means external appearance or visible characteristics of the organism, right? Means whatever visible characteristic, the plant is tall, or the color of the flower is purple, or the color, the seed is round in shape. This is the external characteristic which we can see, right? That is called as phenotype. And genotypes means the pairs of the genes which are responsible for the visible characteristic. That is called as genotype. I hope you have followed.
Yes, hope so. और किसी की कोई डाउट्स है? Yes, Anamika Verma.
Yes, Aramika. Anamika, you can unmute yourself.
Okay. I think teachers नहीं है डाउट्स कोई. हेलो टीचर.
Hello.
हाँ यस टीचर, कोई डाउट्स नहीं है आई थिंक बच्चों के अभी.
Okay. So we'll stop here
Sir,
Yes.
and we'll continue with the same lecture on Monday.
sir,
Yes teacher. Yes,
sir.
yes teacher.
Okay, then we'll stop here. So
Yes, yes.
all of you stay home, stay safe, and study at